Should I go ahead and start?
Yeah.
Okay.
Take it away. Thanks.
Okay, so I'm so pleased to be here. My name is Sarah Nicholas, and I am a beamline scientist at the National Synchrotron Light Source 2. And I can't advance my slide. There we go. So this is me during COVID, and I wanted to say that that's over, but actually we're back to masks. We're still masked up at work. So the National Synchrotron Light Source 2 is a synchrotron light source at Brookhaven National Lab on Long Island in New York, which in the United States. So I'm not in New York City, I am out on Long Island, which is a little east of the city. And here's an air view of Brookhaven National Lab. And there's some features on here. You'll notice this big circle. This is not the synchrotron. This circle is the synchrotron. So the synchrotron here is only about half a mile around to walk it. And this is a different collider accelerator that is much larger and underground, called the relativistic heavy ion collider. But this is where I work, and at Synchrotron Light Source 2. And so you can see cars and things to get some scale. And this is the old synchrotron here, which has been repurposed as a new building. And I'm also showing the Center for Functional Nanomaterials, because that is, as well as National Synchrotron Light Source 2, the Center for Functional Nanomaterials is also a guest and visitor facility. So the synchrotron and the Center for Functional Nanomaterials exist for people to write science proposals to come to our facilities, and we help them run their experiments. So we don't have, our main program of research is to help people complete their programs of research. And synchrotrons, this is a picture of the Australian synchrotron, but I thought it showed the parts really well. There's a linear accelerator, a booster ring, and then, and this is, so the linear accelerator creates the electrons that go into the booster ring. And the booster ring accelerates the electrons until they're reaching very high speeds, and then it goes out into the storage ring. And so this is, I have a similar, this is the one showing inside the Australian, was very similar to what we have. So what is a synchrotron? A synchrotron is a facility that produces tiny beams of very bright broadband light. So we have X-rays, ultraviolet, so going up in size, X-rays, ultraviolet light, visible light, infrared light, and then microwaves. And to get the idea, so these are the brightness and the broadband spectrum are unique features of synchrotron light. And this, oh gosh, I'm sorry. That video, here is a video. Cannot play media. This is a cartoon that is meant to show how electrons are accelerated. And I think I would have to leave sharing and go into my browser. So perhaps we could do that at the end of the presentation. So the broadband color spectrum, and this is in the opposite direction, whereas before they were, the wavelengths were getting larger from left to right. Now they're getting they're getting smaller from left to right. Radio waves, microwaves, infrared, and then here are some objects on those scales to give you an idea. So microwave, approximately, within the range of the size of a honeybee. Infrared, more like pinpoints. Visible light, these are very small wavelengths, 10 to the 6th. Ultraviolet light, 10 to the 8th. And then we have X-rays that are 10 to the 10th meters, and gamma rays, which are smaller still. And we don't make gamma rays. We only go as far as X-rays. Oh, sorry. And so the reason we want these tiny wavelengths is to see very tiny things and to resolve very tiny things like molecules and atoms. The unique thing about synchrotron light is the brightness. We just have a lot of photons in a small area. We call this the flux. But the brightness and the brilliance of a synchrotron relate to how many photons you can get in an area per unit of time. And so here is a light bulb, that's 100 watts. Here's a gas, and this is I think an argon laser, 5/1000th of a watt. So this translates into, a 10,000th of a watt per millimeter squared. There's a little more coming with the laser, but when we get to the synchrotron, we're talking about 30 watts. So 3,000 watts per square millimeter. So a lot more light in a spot. And this is important for being able to look at very dilute samples where there's just not a lot of the element of interest and very small items. So what we do with synchrotron light are different experiments related to absorption, scattering, and diffraction. And these are properties of the way light interacts with matter. These are the principal ones. And you will know some of these already from perhaps work you've done with X-ray diffraction at benchtop scale instruments. But even with your eyes, you can observe these different properties of light interacting with matter. Light is absorbed by matter, and it gets, you can see that if something gets hot in the sun. Scattering, you can see light reflecting off of things. In diffraction, where light is shifting, you may see it sort out a spectrum when it goes through a mineral. So these are all things that you can even see with your own eye and with handheld lenses and things like that. So some of the research that goes on at our synchrotron, a lot to do, surprisingly, with, or maybe not surprisingly, with biology and materials. There's a lot of material science, but there's also a lot of biology and a fair amount of soil science and geology. And I have started the program, or I have, is my job to coordinate the program with marine science at the synchrotron. In geology and environmental science, we do a lot with like environmental remediation. There's actually a surprising amount of planetary science work going on. This is an image of phytoremediation. So what we see here is a metal hyperaccumulating plant. And there you can see how the distribution of metals in different parts of the leaf of the plant. The X-ray crystallography people do a lot with proteins and drug discovery, and a lot dealing with medicine, in particular diseases where there's a structural change that can be observed in cells. So, but my work is mostly X-ray spectral microscopy, and X-ray spectral microscopy is the analysis of the interaction between matter and the X-ray portion of the electromagnetic spectrum. And microscopy is a technical field that's using microscopes to view objects that are not within the resolution range of the normal eye. So this is a picture starting at the top of my beamline. This is where the X-rays come out of the storage ring. And this is where people write proposals. And when those proposals are successful, before COVID, they would all travel to the beamline and collect their data. I would be there to help them prepare their experiment, show them how to collect the data, and stay with them and help them collect the data and analyze the data. And visits are typically 3 to 4 days per cycle, and people stay at our dorms on site. And with COVID, we've had a lot more of people sending their experiments in with instructions, and either I communicate with them. Sometimes it's all clear from written instructions, and other times we'll have several interactions over the course of the beam time to make sure to report the data so far and to make sure that everything is going on track. And some beamlines allow the users to interact, to run the whole beamline remotely, and the person here at the facility just puts this, just loads the sample, and then, but that's quite extreme, and not all the beamlines can do that. So XFM is my beamline, and it's the most beautiful. It's the most elegant, and it's the friendliest beamline, I think, at NSLS2. So this is a slightly larger group than you would usually have. It's usually 3 or 4 people coming to visit. So our beamline at XFM is an X-ray fluorescence microprobe, which means we use X-rays to probe the chemistry of materials, and that's the X-ray fluorescence part at the micron scale, and that's the microscopy part. We're going to talk a little about biogeochemistry, which is for a microscopist all about interfaces and gradients in materials. And I will describe a little of how X-ray fluorescence microscopy works. We'll look at the parts of a beamline for spectroscopy and the parts of a beamline for microscopy. And then I have an example case to show of iron, copper, and sulfur speciation changes of some hydrothermal deposits from Endeavour Ridge, Juan de Fuca, which is in the northeastern Pacific Ocean. So we're a hard X-ray microprobe, and we use it's a kind of X-ray microscope, and we resolve chemically distinct features in heterogeneous samples. So this image over here is a soil nodule, and this is accreted from soil solution around a mineral grain over time. And you can see that we've gone ahead and put false colors on the different elements that we can see. So with manganese in red, copper in green, and arsenic in blue. And so you can kind of imagine, you don't have to imagine because we've measured them, but if you think about the changes in the soil solution and the saturations that happen to trigger the changes between these different mineral layers, that is what the person working on the paper for this would write up when he got home. This was work done by a student at the University of Delaware. So we do that with imaging. We keep the energy static and we move the sample back and forth in the beam. And then after we talk a little more about imaging, we'll talk about spectroscopy. But here are the basics of X-ray fluorescence are, we focus the sample in the path of the X-rays. So we're not able to move the X-ray beam. There's a lot of mirrors and things that once we get them set, we leave them. But so we have stages that move the sample. We illuminate, which is how you use most microscopes. You move the sample, you don't adjust the position of the microscope. And then our detector picks up the X-ray fluorescence spectrum of that spot. So here we have an example where I have put the X-rays here on this red dot. And if we go over and look at the spectrum, we see that there's a lot of calcium fluorescence, there's a lot of iron fluorescence, there's some nickel, some zinc, and some arsenic. And this big peak is the energy I applied to the sample in the first place. So this, for those of you that use SEM EDS, this probably looks not unlike the spectrum that you would get off of that. And the main difference is that instead of applying an electron and reading an X-ray, I'm applying an X-ray and reading an X-ray. We're a hard X-ray microscope, and we can resolve these chemically distinct features in heterogeneous samples. And then this is the basic physical process of X-ray fluorescence. An incident X-ray interacts with a deep core electron. So if you think of the Bohr model, you would be thinking of the electrons on the inner rings of that Bohr model. But here's a 1s. So here we have an X-ray coming in. It knocks out a 1s electron, or it excites this 1s electron. The deep core electron, it propagates out of the atom and it leaves behind this core hole vacancy. And 1 or 2 femtoseconds later, so a very short time later, a higher lying electron decays into the core hole, but it has too much energy and it has to emit a photon. And that photon is an X-ray. And that is what we're actually measuring in our detector, the escaping X-ray photons that are coming from this energy adjustment as electrons move to occupy core hole vacancies. Each element has a characteristic set of excitation and fluorescence energies. And here we're looking at the data for iron. So iron has an edge at 7112. So that's 71,012 electron volts. And it fluoresces at an energy of 6,405 electron volts. And so if you look at that spectrum, you can bracket then the part of the spectrum so this is the light that's coming back off the sample. And we can look here and see, this is the light that is coming from iron fluorescence off of this sample. And we have contrast, so we're able to see lots of different elements, and depending on how heavy, as they go up in Z, we may not be able to see the inner shell electrons. Instead, we may be only able to excite the outer shell electrons, which are at lower energies. So that's why so we have different, so we're pretty good in the sort of transition metals. And then as we get out like into the actinides, that becomes more difficult, because we need a lot more light to fluoresce those things. And for these in blue and in purple, this is what we're working on, where interestingly, we can go that low in energy, but the problem is that argon in the air absorbs the fluorescence coming off. And so by putting the samples in a helium environment, we exclude the argon and we're able to collect fluorescence off of them. So these are the parts of an X-ray microprobe. This is sort of a cartoon of my beamline. We have X-rays coming out of the storage ring, so where all the electrons are circulating, through a 3-pole wiggler to excite them more. It goes through what are called white beam slits, and this is to block some of the light comes through, and then it enters in. So at this point, it's left the accelerator and it's coming into the beamline, and we use mirrors to focus it. We use slits to pinch it down. And we keep focusing it. And this is an important part for spectroscopy. This is a monochromator, and this is where we're actually able to tune the energy of the light. So that's another big difference between what you might see with a synchrotron spectroscopy and what you might see with an SEM, is that we're able to really keep that at a monochromatic light coming onto the samples. So here's more slits, and we focus the sample. So this is all sort of the photon, this is all the delivery of light to the sample. And then from here, this is the measurement of the fluorescence coming off of the sample down here. So I should have said, so this is getting the light to the sample, and then this is getting the light out of the sample, getting the absorbed and excited light out of the sample. And so using these methods is really interested in biogeochemistry, because we think a lot about the cycling and transformation of elements in natural and constructed environments. And this is an example of sulfur redox cycling in marine sediments. So another thing that makes synchrotron spectroscopy different from using something like a broadband spectrophotometer is that we can actually distinguish between different species. I just realized that I skipped that important part early in the talk. These are excitations of different species of sulfur when we change the energy. And the thing that makes that significant and important is that you can actually see different types of sulfur in the sample. And so, which is an important difference between something like an SEM. So we can see sulfides and sulfates have slightly different excitation. So depending on what the chemical species is of your sample, you can see different kinds of fluorescence coming off of it. I am very sorry I didn't say that earlier in the talk. So when you get to an actual field site where you want to look at the speciation of something, by being able to distinguish the different chemical species of an element, you can try to work out what these transformations are happening. And this is a cartoon showing sulfur speciation at depth. This is from Jorgensen 2019. And so you see a lot is going on with sulfur, from sulfate in the seawater and in the top of the sediments. And then we start to see reduction and we have anaerobic methane oxidation, we have reduction to hydrogen sulfide. And so these are all processes that in the solid phase that we have an opportunity to see if those are there in a sample by doing microspectroscopy on the sample. So we might discover pyrite, for instance, at the extreme end of reduction. And then we might still be discovering sulfate minerals also in that, farther up, closer to the oxidized seawater in that same sample. So sulfur is an important element, and we're very interested in sulfur. Here's another example from sulfur looking at hydrothermal event, where we're looking at transformations and speciation of iron and sulfur as seawater comes in and mixes with hydrothermal fluids. Solids are formed. Some of them precipitate as part of the chimney, some of them go out as particles. But we see these different redox reactions are happening with respect to sulfur and iron in this system. So looking at these chimneys, I'm going to be showing you an example of these chimneys. So this is a hydrothermal spreading center, and as the older chimney moves off its source and becomes dormant, an interesting question is what happens to this structure? What happens to the structure itself? And what happens to the chemistry and the mineralogy of that structure as we move the sample? So here is a figure from Meg Tivey, the 2007 map, and these are the some hydrothermal vent locations. I'm going to be showing an example of work I'm doing here at Endeavour Ridge, which is part of Juan de Fuca. This is about 200 miles west of Vancouver in the Northeast Pacific Ocean. So this is the first science commissioning that we did for looking at sulfur at my beamline. These are chimney samples from Endeavour. So this is a figure from Katrina Edwards' paper on the Endeavour Ridge. And this is a cartoon. This is actually not the same submarine that sampled this sample. This is a picture of the Alvin. We can see coming in and actually going to a live chimney, knocking off a piece and taking that away is one way to do it. And so you can see here, they're also sampling the fluids. but it's possible to go to a live vent and take a sample and have sort of a time zero look at what is happening at a hydrothermal event. And in the case of Endeavour, in addition to that live vent, by following, you see here, this is the spreading, the axis of spreading. And if you go normal to that, you should be encountering older and older samples that have moved farther and farther off of this spreading center. And this is a transect that I have samples from, and I will be showing some of those today. So these are here, there's the live sample and then the subsequent older samples. As if you imagine the plates are moving, the hotspot is staying in the same plates, the plates are moving apart and those chimneys become stranded. So we're interested in those. We have 6 samples from the hydrothermal chimneys. One is the live one, and one is 5,200 years old. So on the right, we see the live sample. And on the left, on the left, you see the live sample. So this is the fresh sample that's just formed. And on the right, you can see the sample that has aged. And a big thing we notice in the difference between these samples is the chemical distribution is different. And in particular, the amount of copper. There is less copper in the older sample. And everything is more oxidized. You know, it looks a little fuzzy, it looks a little grainy. It's less equant that you see here, where you can see distinct mineral grains. So that's something going on with the aging of the sample. And if we look at a very small sample and start to take points on it, we can see these different speciation of sulfur within the sample, where we have sulfate, and then we have 3 different sulfide minerals within the sample, a copper, an iron, and a mixed copper and iron sulfide in the sample by looking at these little spots. Another thing we can do is, instead of holding the sample in one place and changing the energy to get a spectrum, we can hold the energy at different points on that spectrum. So we select energies based on the spectra we observe, and then we take whole maps at those energies and integrate those maps. And when we do that, and then we fit them using linear combination fitting, which is a linear algebra method, we discover that we can sort of map the distribution of elements in the sample. So in this case, here are places where there is sulfate in the sample, and then 2 different sulfides. And you can see their distribution is slightly different within this sample. These are new slides that I took this year. I was very excited about these. I got the original samples. They had been done by a graduate student, and the thin sections were good. And I was looking at them. And then I had occasion I asked for the original hand sample because I was interested in putting them on a different substrate. Because I wanted to do diffraction. And so I wanted to know the mineralogical combination of the substrate, and which is not very easy in glass. And the great surprise to me was when I got these samples back, then these are just new preps of the same samples. All these structures that had been lost in the first preparation were present in the second preparation. And I am super excited about these because these seem to be microbial exudates on the outside of the sample. So you see here, you can see the sulfide, gleaming. This is a light microscope image, but you can see that shine of iron sulfide. And then this brownish material, when we look more closely, we can see that it's like little bubbles, and these bubbles are iron oxide. And I believe these seem to be microbial iron oxidation reaction that is producing these exudates as a footprint of that microbial oxidation. And if we look at farther on, we can see, so here, this map shows calcium also, and we see some kind of a carbonate out here, some kind of iron carbonate, and then moving more into the sulfide part of the sample. Now the problem so we can combine that. So here we have the X-ray fluorescence. We've done some spectroscopy. We can also use diffraction on these samples. And here is that same, here is another edge of a sample where we have an oxidized rind moving into a sulfide bearing base. And we can take, so here's the hand sample, the thin section and you can see the sulfide there. It's very shiny. Down here, the oxide, which is brown and dull. And if we go ahead and take diffraction patterns across it, we see poorly crystalline iron oxides moving into more crystalline iron oxides. Here's an empty spot where I can just see the quartz in the slide. And then we start getting into marcasite, sphalerite, and pyrite. So that would be the primary sulfide mineral that was precipitated at the time the time the vent was active. And these are subsequent oxidation products on the edges of the sample. So this is just an example of a marine application for synchrotron microprobe. And there are plans for Africa's first synchrotron. This is something that it's being discussed. A budget is being put together. And then some work that I think is important that's being done is that there are some traineeships and postdocs available for African postdocs and students to study at the European synchrotron. So this is the European Synchrotron Radiation Facility, which is in Grenoble. So that is one of them that's active. And so this is an exciting process to think of starting a new synchrotron facility. There are synchrotron facilities in Europe and Asia. There's one in South America. There's one in Australia. There's several in North America, but this would be, and there's one in Jordan on the Arabian Peninsula, but the whole continent of Africa doesn't really have access to this kind of large research. And so it's exciting that a plan is being formed to build one. Thank you. I am sorry that was so rushed. Does anyone have, this is a marine dinosaur. This was measured at my beamline. This is the yttrium signal. And what's interesting about the actual sample that if you hold the sample in your hand, it's about the size of a cell phone. And you can't see you can only see a tiny portion of the skeleton. All of this is hidden under shale rock, but is revealed when you use the X-rays. Does anyone have any questions? I can't see the people. Oh my gosh, that was faster than I thought it needed. So I did not mean to go that fast. And I would be happy to go back if you want to look at any other parts. And I'd be happy to answer any questions.
Did you want to try to load that video too?
Oh, yes, that's a good idea. Okay, so I'm going to unshare. I'm going to unshare here. And I'm going to go to my, stop share. Okay. I very unfortunately, I had this meeting beforehand where I had to restart my computer and I lost a lot of stuff. So I think now if I share, can I select my, okay, now I've lost my Zoom window. I'm so sorry. Okay, here we go. So, oh, that's the wrong video. Are you guys seeing my screen?
Yeah.
Okay, my link to the video is gone now. I'm really sorry. Let's see what we can. Okay. I'm going to copy it out of the talk. Somehow I'm still in my talk.
Oh, I think you have to exit out of slide presentation mode in your PowerPoint.
Okay, here's the link to the video. So I'm going to copy that into my browser. And I'm going to put that here. And now I'm going to share Here we go. That's not it. Can I send you the link, Wynn? That might work better if I put it in the chat because I'm not able to pull it up.
Yeah, I can try that. In the
I'm-
meantime, it looks like Comfort has
Oh, okay.
questions. So
Hi, Comfort.
Oh, thank you very much. And I find the lecture very interesting and exciting. I congratulate you for the depth of work that you have been doing.
Thank you.
I think it's really a lot, and I wish you more successes. I just wonder whether you have any arrangements or any window for exchange programs or visits between institutions, that maybe have some students. And another question actually is, how do you talked of some graduate students and postdoctorals with you. How do you get them in? If people are interested, anybody interested, what is the procedure or the process of getting to, make the most, getting to, interact more
Yeah.
with your laboratory? Thank you.
Well, our lab, I have reached out to a couple of people with that because the UN Year of Science will be in September, or the UN meeting. There's a 2-week meeting for the United Nations on science, and part of that is coming out to visit here. And there's a whole section on the African light source, which is being proposed to be built, but of course it hasn't been built yet. The European synchrotron radiation source in Grenoble has some exchanges. These are traineeships for African students and postdocs, and that'll vary from what field of science they want to do. There's a lot of work being done on protein crystallography, and that is especially important for people interested in medicine and disease. And in terms of materials side, it's sometimes hard to explain to physicists the difference between geological materials and materials. They don't find that to be that interesting a difference, but there is a lot of geological work that also goes on at the ESRF. But the main way people get to come to the beamline is by writing a proposal for a sample. And within Europe, these are generally not only, so your research is always free. Once you get to the synchrotron, we don't charge you for consumables. We don't charge you for your time. You're here to do your experiment. Within Europe, they also typically pay for travel within participating countries in Europe. And in the US, the main way to get time on a synchrotron is to write a proposal. Then you would typically be writing it from a university. And so the proposals are fairly short, but you would include a list of what the samples are. And the main thing is the science reason for why that sample should be analyzed. And it typically includes a lot of data on the measurements that have already been done, like light microscopy, dissolution chemistry, that kind of thing. But I reached out to our group here about traineeships and fellowships specifically, and I have not heard back, but I know there are some within the European synchrotron system. And the closest synchrotron to you in Nigeria, the closest synchrotrons are probably the ones in Europe, but there is also Sesame is a new synchrotron, a fairly new synchrotron that was built in Jordan. And it is recycling of a synchrotron that they were taking apart in Berlin when they built a new one. They took the whole accelerator over and set it up in Jordan. And now they have, I think, 14 beamlines running, and they're collecting a lot of data, which is really important for it. So it's important to have a regional facility. Another example of a good regional facility that started out as quite a small synchrotron is the Thai light source in Thailand. And they took the accelerator from a Japanese synchrotron that was decommissioning and built, and there's a whole dynamic science program going on in Thailand, where it might be quite difficult for those scientists to get time in China, where there are a couple of synchrotrons, or in Japan, where there are a couple of synchrotrons. But at the Thai light source, it's really serving the community in Thailand, Laos, Vietnam, the Philippines, Indonesia. So having a small facility is working really well. But I think the traineeships are exciting for me, so that when you have an African synchrotron, you will have African staff ready to run it. You won't have to bring in people from away.
Great, thank you. Yes.
So.
The animation is running for me if you want me to.
Oh, could you show it? Can you share? I'll stop sharing.
It's the overview animation, right?
Yes.
Okay.
So.
Do you see it?
Yes.
Okay, cool.
So this is meant to be the flight source in the UK called Diamond, and we're starting on the outside. It does look approximately like this. That's the support building over there. So the thing that looks like a big donut is the synchrotron. And now we're sort of taking the roof off. And if we start in the linear accelerator, the electrons are accelerated to a ground here, like you would do in a lot of instruments, but much, much larger. And it goes into this booster ring. And so, but they're changing the direction and accelerating electrons in a ring. And when they reach a certain speed, then they go out into this big storage ring. And what you're seeing are experimental hutches lighting up, and that is meant to show the X-rays coming through the hutch. And so whenever you change the path of an electron, it loses energy. I would say now we're the electron shooting down but whenever you change the path of the accelerated electron, it has to compensate for the loss of energy, it has to emit a photon, and that photon is an X-ray. So by having the instrument set up tangent to the ring, you can run a lot of experiments. And although the cartoon shows them as distinct flashes of light, there are instruments where we can measure how far apart they are, but it's nanoseconds, femtoseconds apart. So for most experiments, you are not aware of any separation of your X-rays coming in. So Comfort, will I be able to find your email through the registrants? Because I'm expecting to hear back from a couple of people in the US who are working with the African program. And I could pass your, but you'll be listed on the registrants.
We can send in, once you have that information, we could send it out to everybody.
Oh, okay.
So they had that.
Thank you very much.
Oh, sure.
Yeah. And were you able to join the Slack, Sarah?
Oh, oops. No, I haven't joined. Is there a Slack?
If you want to be a part of it, that gives us a way just to interact over the course of the week if people have questions.
Okay, I will get on that. I
want to interact with you.
Yeah, we have that here too. It's just the meeting I had just before this, I had to restart my computer. And so everything that was keyed up has unfortunately been lost. So it's interesting because it's such a, oh, do we have to wear special suits? So the way we protect ourselves from the X-rays is that all the experiments are done inside these shielded hutches. So there's lead, and depending on how strong the X-rays are, there's lead and stainless steel, or there's many, many layers of lead. And so what we do when we run an experiment, I should make a video of this. The main thing that happens is you can't be there when your experiment runs. So you set up your experiment, you put your sample on the stage. If you're, sometimes people react samples, and so they may have some apparatus going, but you set up the sample and then you have to close, we call it searching and closing the hutch. And when you search the hutch, it has to be just one person so that there can be no confusion about who did what. And you look all through that. So the hutch is the smallest experimental hutch I've ever worked in was about 2 meters by 2 meters. And the largest one is about 4 meters by 3 meters. And at about 3 meters in high. So there's a volume to that. But just one person, when the experiment is all set up, your whole team leaves the hutch and just one person looks all around the hutch to make sure that no one is in there. And then you start a search, which is where an alarm goes off and a light flashes, and you have to push a series of buttons confirming that there's nobody in there until at last you close the door. And only when you have confirmed that no one is in there and close the door, and you have to be on the outside of the hutch holding the door, holding the button down to close the door, only then can you open the shutter and let x-rays into the hutch. And that's how we keep ourselves safe from the x-rays. And other than that, the precautions are sort of basic laboratory precautions, closed-toed shoes, safety glasses, that kind of thing. So, are there any other questions? Do some of you do microscopy now, light microscopy or electron microscopy? Oh, okay. So, all right. So I think what I should, oh, thank you. So I think maybe what I should do is just join the Slack channel, and I'll sort of keep an eye on that during the course of the week. It's about 11 o'clock in the morning here, and I guess it's the afternoon for you guys. So I hope you have a nice evening, and I'm glad that you're able to be there together. I guess there's the virtual and the in-person component, but my hope is that next year this can be, we'll all be together. So thank you so much. So I think I will leave the meeting.
Yep, go ahead. We'll post a recording of it if anyone wants to go back and revisit anything, and then play that.
Okay.
As long as you're comfortable with that.
Well, I wish you'd post last year's because I didn't mess it up so much.
Oh, I think we did have last year's too.
Oh, okay.
Maybe already up there.
Oh, okay. Well, thanks so much. I was just, I said, and now you have to restart your computer. And I thought, oh no, don't do that. Okay. Thank you very much.
Thank you.
Bye.